Okay, hello, welcome to this video lesson on the nucleic acids topic. It's November, it's back in some form of lockdown again, and we're doing this all over again. So this is going to be the first part of two-part lesson on the structure of nucleic acids. In the first part we're going to cover DNA, its structure, how it bonds together to form the famous double helix, and in the second part we're going to look at RNA, ribosomes, and a little bit of the history and some of the people that are involved in the experimentation that led to this sort of groundbreaking, most famous of images within biology. Okay, so moving on, our very first thing, right? This is our first lesson, first part, the structure of DNA. So here's the spec. You are expected, just like all other biological molecules in the A-level course, to know the structure of a nucleic acid. You're expected to know how it bonds together, how they bond together to make a polymer. You are expected to know that everything in life has some form of nucleic acid. Mostly we're talking about the famous DNA double helix held together by hydrogen bonds. Over here we can see the structure that you are expected to learn. Notice it's not as complicated as the structure of an amino acid, where you're supposed to know the full chemical structure. You only have to know the three parts of this for A-level. However, I am going to show you the full chemical structure because I think it makes it make a lot more sense as to why DNA is called what it is and how it works the way that it does, and the difference between DNA and RNA. So first things first, I don't know if you know this, it's good pub quiz knowledge. What does DNA stand for, right? And what does RNA stand for? Well, DNA stands for deoxyribose nucleic acid, okay, and RNA, just we simply drop the deoxy bit, which should tell you something about its structure. Okay, so deoxyribonucleic acid and then just ribonucleic acid. You're probably thinking that the difference is going to be something to do with an oxygen, and you'd be right. Okay, so moving on, why is this the case? Well, nucleic acids, they form weak acids in solution. Okay, so they're called nucleic acids. Ribose, because that's the sugar that they're made from. Okay, so ribose is like glucose, fructose, galactose, whatever it is. Deoxy or not is the presence of an oxygen at a critical point on that ribose sugar, particularly carbon 2. We'll get to that in a bit. Okay, these are another example of a polymer, just like the carbohydrates you will have learnt about and the proteins you will have learnt about. They are polymers. They're composed of monomers. The monomer is called a nucleotide. Okay, the polymer is of course our DNA strand. Okay, our DNA or RNA strand. Okay, so let's do the structure of a nucleotide. I'm going to do this nice and quickly, obviously. They've got three parts to them. First one is the phosphoric acid, or a phosphate group up the top there. Okay, so you've come across phosphate groups in biological context before, in terms of phospholipids, various other places, ATP, the phosphates there. So we've got phosphoric acid at the top there. That little OH group should be making your little biochemistry brains think, ooh, what sort of bond could form there? We've got this deoxyribose. Okay, this is DNA we're talking about it, and there it is. Now you should be able to see from this, it's quite clearly a pentose sugar. 1, 2, 3, 4, 5 carbons, pentose, as opposed to glucose, hexose sugar. The oxygen's at the top, and down here, look, if you compare this to what you know about the structure of glucose already, you've got an OH group here, and you'd expect there to be an OH group somewhere on those two there, but there's not. Right, that oxygen has gone away, and it's deoxy. Okay, it's an oxygen missing over there. Now, of course, because it is those oxygens, right, those OH groups where condensation reactions happen, might go some way to explaining one of the properties of DNA, which is that it's less reactive than RNA, which is very useful. Okay, so we've got the phosphate group, we've got the deoxyribose, and then we've got the all-important bit on the end, which if you were paying attention in a very, very short bit of this at GCSE, you probably did. We've got a nitrogenous base. Now we'll get into those a bit later, but they are the ones that have the letters A, T, C, and G, although for A-level purposes you actually do need to know what they're called. Okay, so we'll come to that in a minute. So, the bases. There are differences here between DNA and RNA as well, and we're not going to go too much into RNA, but for DNA, you've got the special base thymine, T, which is only found in DNA, and for RNA it's uracil. There are only 4 at any given time, because of course they have to pair. Okay, that's notwithstanding any weird epigenetic stuff that we're going to come to in year 13. So only 4 different base pairs present at a time. Adenine is what A stands for, cytosine is what C, guanine is G, thymine is T, that's your DNA bases, and then uracil is in RNA. So in DNA we've got A, T, C and G, adenine, cytosine, guanine, thymine, and in RNA it's A, U, C, G, adenine, uracil, thymine, and cytosine and guanine. Okay, so this should look familiar. How do you then put a nucleotide, a series of nucleotides together to make a strand of DNA? Okay, so we've got up here, right, we've got our one nucleotide. Down here on the end, carbon number 3, 1, 2, 3, and I'm mentioning these carbon numbers because they become important in just a little bit. So we've got carbon 1, 2, 3, you've got the OH group there, and then you've got the OH group on the next bases, the next nucleotide's phosphate, okay, and that's nearest to carbon 5 here, so that's the phosphate group attached to carbon 5. They bond together in what should now be a familiar structure. They form a condensation reaction, okay, they have a condensation reaction, and what we're looking at here, this is called a phosphodiester bond, and of course, like any other condensation reaction, it gives off water, okay, a molecule of water there, okay. Now it only ever happens this way round. It only ever happens phosphate group to 3' end, okay. So you get one phosphate, you get one whole nucleotide up here, another one comes along, binds the phosphate group to the 3' end, and that is how a DNA strand knows sort of what direction it goes in. That's how the enzymes are adapted to fit these two things together, and it's very, very important in how DNA is replicated and how it's read that that should be the case. So DNA always goes in this direction. It always attaches the 5' end here with this phosphate to the 3 carbon end there. You never get one upside down, okay? That's called a phosphodiester bond there. So you are expected to know that is a phosphodiester bond, just like you're expected to know glycosidic bond and peptide bond and ester bond. It's just another form of biological bond you're supposed to know. So we're going to zoom out a bit now. Okay, so coming a bit further away. Okay, we've got then, that's the context of what we've done so far. That's one nucleic acid, and here this diagram here is simplified to what is actually on your syllabus. Okay, with just a blob for the phosphate, right? Pentagon for the sugar, and a rectangle for the bases. Okay, and new molecules always join there down on the 3 end. See the carbon 5 and the carbon 3 are highlighted here to make that point that's always going in that direction. Okay, so on which note, right, it means that whenever you look at a nucleic acid chain, a polynucleotide, they've always got a particular end. They've got a handedness. Okay, there will always be an end where 5' end is sticking out, where the one with the carbon 5 is sticking out the top, and there'll always be an end where carbon 3 is sticking out the bottom. Okay, you'll never find one with 2 carbon 5 sticking out out the end, or 2 carbon 3s. Okay, it gives DNA a handedness, and that's incredibly useful, because if I picked up any book or any bit of text, the first thing I'd have to know about how to read it is where to start reading. Now in English, of course, we start at the top left and we work from left to right, and then we work down the rows. Okay, that's not the case in every language. So the first thing you need to know about any code is where the start is. Okay, is where you start and which direction you read, and that's what this 5' to 3' system, as it's called, gives you. There'll always be an end where the ribose is sticking out, the deoxyribose is sticking out, and there'll always be an end where the phosphate is sticking out. And DNA is always built from phosphate down to ribose, from 5' down to 3', and that's also the direction in which it's read. Okay, that's also the direction in which it's read, 5' to 3' and replicated. So, the structure. Of course, it's double-stranded. You don't just get one strand, so now we're building up, zooming out a little bit more. It's double-stranded, and actually the strands don't run 5', 5', 3', 3'. They run 5' to 3' and 3' to 5'. So this one is back to front. So they're not parallel like that, they are anti-parallel like that. And that is not your mum's northern sister called parallel. Ooh, me anti-parallel. Right. So two double helix then, once they're anti-parallel like that, they then twist and rotate together into the now really, really famous, you should be able to see this everywhere, but anytime a BBC newsreader says the word science, there's some sort of graphic of this picture, the famous double helix. Okay, so we've got two strands coming down, so I'll follow this one down here, follow it down there. You've got this strand coming up this way, like that, and in between you've got the bases. Now the bases stick out like the rungs of a ladder, and they are the ones that pair up. And if you remember your GCSE, particularly in detail, A binds to T and C binds to G. The reason for that, complementary base pairing. It's just about structure, just like anything else in biology, it's about shape. Okay, so on base pairing, they're held together by everybody's favourite bond, hydrogen bonds. Okay, and they form these called base pairs, A with T, C with G. They are specific, you do have to remember them. A binds to T and T with A, and C binds to G. And the way I always remember that is A and T spell at, the word at, and C and G are the initials of the guy that taught it to you. So that's very important. Right, so what that means then is if two strands can bind together nicely, right, they're going to have matching pairs. So one side is not going to be the same as the other, but is going to be able to match up, join up, and form those hydrogen bonds. So if this side was all A's, this side would be all T's. If this side was alternating AT, 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 this side would be alternating T, AT, AT, A. Don't be tempted to write in an exam of any sort they have the same sequence, because they don't. The same sequence wouldn't fit together. They have complementary sequences. Okay, they are called complementary base pairs and complementary sequences. Now, of course, that means the code is different on each strand, and therefore only one can be used to code for a gene. One is used to code for a gene, and the other one is there purely for replication, so you can make more copies when it comes time to do cell division. You can make more copies of the strand that's going to do the coding. We call these the sense strand. That's the coding strand, the one that actually codes for protein. We call that transcription. That happens there. The other is called the antisense strand, and that is used for replication, as it says there. Okay, so keywords, sense and antisense, or coding and non-coding strand. Okay. If we want to go into a little bit more detail, now you don't have to know the structure of the bases. Okay, you don't have to know those at all, but it might help you to understand, particularly for those of you who are also chemists, that the reason they only fit together, as you can see, we've got 2 hydrogen bonds forming between A and T, and we've got 3 hydrogen bonds forming between C and G. Okay, that's why they bond together so nicely. Okay, also it's a size thing, it's a size and shape thing. You might notice in each pair you've got one double ringed base, A and G, and you've got one single ring base, T and C. Okay, now that's why they then fit nicely together and lead to a nice straight, evenly spaced double helix. Okay, so if we were to look at this one, we could quite easily then work out, and this might be a standard easy peasy AS level question, what the other bases would be opposite G, T, C, C, and we would say they're going to be C, A, G, G. If we were asked to put the prime numbers on, what carbons are sticking out, well, on the left there we can see that that's the 5' end, because the phosphate's sticking out, and that's where carbon 5 is sticking out, and down on the other side we've got the 3' end sticking out, because they are anti-parallel. Okay, down that end of course then is 5', and that end must be 3'. Okay, good. So we'll finish here for the first part of this video. This basically just shows us a little bit more detail and leads us into the next one, that how is it that we can just swap a base out in RNA? Well, if you look at uracil down there in the bottom right, uracil's got a very, very similar structure to thymine. So in terms of forming hydrogen bonds, it can basically do the same thing. Okay, basically do the same thing. In fact, the only difference the eagle-eyed chemists of you might notice is that methyl group, right? That methyl group that's just up there, okay? That methyl group that's just up there on thymine that isn't there on uracil, okay? And that's how uracil so neatly swaps into thymine and bonds with adenine, okay? And that's a really quick run through your first lesson on the structure of nucleotides, the structure of DNA, and we'll come back for part 2, which is on RNA, ribosomes, and a bit of the history. See you then.